I'd like to welcome all of you to today's webinar where we'll be discussing the prevalence of nitazene analogs. My name is Dr. Andrew Holt, and I'm a member of the Aegis clinical team. Today, we will discuss previously published findings on nitazene analogs, some recently published information regarding their metabolism and potency, and trends in Aegis test results over time. Nitazene analogs belong to the 2-benzyl-benzimidazole subclass. They're structurally different from opioids, but they do attach to the same mu-opioid receptors as the more familiar opioids like morphine, buprenorphine, fentanyl, and others. Over time, they have demonstrated a significant potency and risk for development of addiction. Isotoniazine is believed to be the first nitazene analog discovered in the illicit market, with reports dating back to 2019, originating from Europe, Canada, and the US. Metonitazene was discovered soon after in early 2020 and it surpassed isotonitazene in prevalence to become the most prevalent nitazene analog detected through 2023. Another analog, protonitazene, was first detected in 2021. It became the second most commonly detected nitazene analog in 2023. And all in all, according to the DEA's latest National Drug Threat Report released this year, there have been over 4,300 reports of nitazene to their system since 2019. Nitazenes were originally developed in the 1950s by pharmaceutical companies looking for new analgesics, more potent analgesics than morphine. Numerous compounds in the benzimidazole class were formulated with the intention of creating that new analgesic. As part of these studies, they found the most potent derivative was approximately 1,000 times more potent than morphine. This finding, along with other findings like it, led to a discontinuation of development of the nitazene analogs as prescription drugs due to concern for their addictive properties and this high potency that they were finding. To date, there are no nitazene analogs that have been clinically approved for use from the pharmaceutical perspective. U.S. drug laws are largely based upon the identification of specific agents. And this, in some cases, allows new substances which are developed and introduced to evade legal repercussions for some time. As of 2024, however, 10 nitazene analogs are classified as Schedule 1 controlled substances in the U.S. And when you look at U.S. code and where you're going to find the list of controlled substances, 21 U.S.C. 802 is the section where you would find that, and they outline the criteria of controlled substances here in the U.S. 21 U.S.C. 813 allows for any substance that is structurally similar to a known Schedule 1 or Schedule 2 substance to be treated as a Schedule 1 substance as long as it's intended for human consumption. And that's the important thing to consider, because what happens in many cases is these substances are sold and labeled as not for human use. And that's one way that they try to evade controlled substance scheduling laws. And it sometimes works for a little bit of time. So these nitazenes are relatively new as far as introduction into the illicit drug supply. So there's very limited information available regarding their potency in comparison to fentanyl and other more traditional opioids that we're more familiar with. However, new studies are being developed and are starting to appear in the literature. And one study, which we've referenced here, looks at 14 nitazene analogs and compared them to illicitly manufactured fentanyl, prescription opioids, and other designer opioids as well. These experiments were in vitro, so they were not in vivo studies, they're in vitro studies, and demonstrated that the nitazene analogs are more often than not, they're found to have a greater potency than more familiar opioids. This slide has a lot of information, very complex, but I'll do my best to make it easier to understand. What we're looking at is the potency and efficacy of nitazenes in a morphine beta-arrestin 2 in vitro assay and a morphine G protein coupled receptor kinase assay. There's two columns, the two columns to the right. The EC50 is a measure of potency. And so the lower this number is, the more potent the compound is expected to be. Again, all of this is in vitro. The Emax column is a measure of efficacy in comparison to fentanyl, which is what all of these substances were compared to in these two tables. From an efficacy standpoint, the higher the number, the more efficacious the substance can be assumed to be. So in this table, we've highlighted the substances in bold, which are more potent than fentanyl in the EC50 column and more efficacious, I guess, than fentanyl in the Emax column. So about half of these nitazene analogs are more potent than fentanyl, and about 2/3 of these are more efficacious than fentanyl. So again, the lower the number in the EC50 column, the more potent it is, with fentanyl having an EC50 value of 14.4. So you have some that were significantly smaller EC50 values than fentanyl. And then fentanyl as the baseline efficacy getting a value of 100, you'll see some that are, again, about 2/3 are about more potent or more efficacious than fentanyl, excuse me. Other publications support these findings, which indicate that a significant potency and risk is associated with exposure to these nitazene analogs. One study referenced here concluded that exposure to nitazene analogs creates a high risk of intoxication and overdose due to this potency. And another study referenced here concluded that there's a risk for overdose associated with intentional and unintentional exposure to these substances. And they called for a change in how to manage care during circulation of highly potent novel drugs such as these. We'll now shift our focus to results from Aegis testing for nitazenes. As part of this analysis, we looked at more than 800,000 urine and oral fluid specimens collected over about a 15-month period from January 23 through the end of March 24, where testing for fentanyl and designer opioids was ordered by the practitioner, clinician, healthcare provider who is ordering the testing. All of our testing is done based on medical necessity and at the discretion of the ordering clinician. Samples came from over 900 practice sites, and we only included sites which submitted specimens continuously throughout this entire time frame. And we compared the results of Q1 2024 to Q1 2023, when fentanyl was detected, which equated to 41,323 samples. With looking at these samples, there was a 17% increase in identification of nitazene analogs overall in these specimens. Further analysis, when these nitazenes were identified, fentanyl was detected 99% of the time. So there's a significant co-positivity between these substances. Further analysis of these samples, we saw a 14% decrease in samples with only fentanyl present. So 86% of samples had something more than fentanyl in it, either a fentanyl analog, a nitazene analog, or both. 25% increase in samples with fentanyl and a fentanyl analog. A 33% increase in samples with fentanyl, a fentanyl analog, and a nitazene analog together. So at least 3 different opioids detected, in many cases more. There was a 63% decrease in samples with a fentanyl and a nitazene analog identified. So really what you can deduce from this is that there is the illicit drug supply is extremely complex and changing over time. So our findings show that circulation of these substances is relatively steady, despite most analogs being considered controlled substances in the US. So nitazene analogs and fentanyl analogs, along with fentanyl, relatively steady circulation of these substances, although the substances themselves can change individually over time. New analogs are introduced regularly. It's important for laboratories to routinely adapt your testing menus to allow for detection of high potency novel opioids that are in circulation, as these do change over time and new substances are introduced. It's important to be able to detect these new substances. And while identification of fentanyl and fentanyl analogs does allow for risk assessment, for those individuals encountering high potency drugs, it really only tells part of the story. Remembering that some of these nitazene analogs are more potent than fentanyl. So testing for these substances allows for an enhanced risk assessment. Thank you for tuning into this month's webinar. We hope that you found it informative. If you have any questions, please do not hesitate to contact us and be sure to look out for future notifications of webinars in the future. Thank you.